A research team at the Salk Institute published findings in 2019 that measured cellular repair activity across different metabolic states. What they documented was not incremental variation, it was binary activation. When glucose remained elevated, repair pathways stayed dormant. When it dropped below a threshold and stayed there, repair mechanisms that had been biochemically silent for hours or days switched on within 90 minutes. The body does not repair itself constantly. It repairs itself conditionally. And the condition is the absence of incoming fuel. Most conversations about metabolic health focus on what happens when you eat. Insulin response, glycemic load, nutrient partitioning, those are real. But they describe the fed state. The body spends roughly 16 hours a day in some version of fed or recently fed. Now, what almost no one discusses is what happens during the hours when it is not, because those hours are not neutral. They are not maintenance. They are when the system runs a completely different operating protocol. The standard model treats fasting as caloric restriction with a time component. You eat less by eating less often. That model is incomplete. It describes one outcome but misses the mechanism. Caloric restriction without a fasting window and intermittent fasting at equal caloric intake produce different cellular outcomes. Same energy deficit, different biological response. The difference is not the amount of food. It is the presence or absence of a metabolic signal that tells the cell whether fuel is coming or whether it needs to shift into maintenance mode. That signal is insulin. But insulin is not just a glucose transport molecule. It is a master regulatory switch. When insulin is elevated, the cell prioritizes growth, storage, and division. Anabolic processes. When insulin drops and stays low for an extended period, a different regulatory network activates. It is called autophagy, and it does not run in the background. It runs instead of the other protocol. Autophagy is not a cleanup process. It is a disassembly and recycling protocol. The term comes from Greek, auto, self, and phagy, eating. The cell begins to consume its own damaged components, misfolded proteins, dysfunctional mitochondria, lipid aggregates that accumulate during normal metabolic activity. These are not cleared passively. They are actively tagged, engulfed by a structure called an autophagosome, fused with a lysosome, and broken down into amino acids, fatty acids, and sugars that the cell reuses. This is not speculative. Yoshinori Osumi won the Nobel Prize in Physiology in 2016 for identifying the genes that regulate autophagy in yeast. Those genes are conserved across species. Humans have them, and they activate under the same condition. Nutrient deprivation, specifically the absence of insulin signaling and the activation of a protein called AMPK, which acts as the cellular energy sensor. When AMPK detects low energy availability, it inhibits mTOR, the master growth regulator, and autophagy begins. The system does not activate after 12 hours of fasting in everyone. It activates after insulin has been low and stable long enough for the cell to shift states. For most people, that threshold is somewhere between 12 and 16 hours after the last meal. It depends on glycogen stores, activity level, and insulin sensitivity. But once it crosses that threshold, the repair protocol is not partial. It is systemic. A 2017 study published in Cell Metabolism tracked autophagy markers in human subjects across different fasting durations. At 8 hours, autophagy activity was barely detectable. At 12 hours, it had increased modestly. At 16 hours, autophagy-related gene expression was upregulated by more than 300% compared to baseline. The body had entered a different operational mode, not starvation, cellular maintenance. The implications are not about weight loss. Autophagy occurs independently of caloric deficit. You can fast for 16 hours and eat maintenance calories in an 8-hour window and still trigger the protocol. What matters is the uninterrupted absence of feeding, because the cell does not start repair while it is still processing fuel. It waits. And if you never give it that window, the repair protocol never fully activates. This is where the model of constant small meals breaks down. The advice to eat every few hours to keep metabolism elevated is biochemically sound for preventing hunger and maintaining stable blood sugar. But it is also a guarantee that autophagy remains suppressed. Every time insulin spikes, even modestly, mTOR reactivates and autophagy shuts down. You are not giving the system the metabolic space it needs to run its maintenance cycle. The research on this is not emerging. It is established. A 2010 study published in Nature documented that mice engineered to have impaired autophagy developed neurodegenerative symptoms, accumulated damaged mitochondria, and showed accelerated markers of aging. The system is not optional. It is how the cell prevents the accumulation of biological debris that would otherwise compromise function over time. In humans, autophagy declines with age. A 2016 study out of the Buck Institute for Research on Aging measured autophagy efficiency in tissue samples from individuals across different age groups. The autophagy flux in 60-year-olds was roughly 40% lower than in 30-year-olds. The machinery is still there, it just runs less efficiently. And one of the few interventions shown to restore autophagy activity in aging cells is prolonged fasting or caloric restriction that mimics the fasting state. That is the mechanism the research documents. And one of the more interesting areas of current research is whether specific natural compounds can interface with this pathway in a meaningful way. There is a peer-reviewed body of work on this. Research has identified a natural compound that supports this neural and mitochondrial mechanism at the cellular level. It is not a drug. It is not a treatment. It is a natural formula built from published research on brain and cellular energy metabolism. If you want to understand how it works, the link is in the description. This is a sponsored recommendation and individual results vary. The framework this gives you is not about disease. Standard clinical labs measure pathology, HbA1c, fasting glucose, lipid panels. Those are retrospective markers. They tell you what is already broken. Autophagy operates in the space before that. It is the difference between fixing small problems before they compound and waiting until the system is visibly damaged. Most people never access this because they never stop eating long enough for the cell to shift states. 3 meals and 2 snacks keeps insulin elevated from morning to night. The body stays in growth mode. Repair stays off. And over years, the accumulation of damaged proteins and dysfunctional mitochondria begins to interfere with normal cellular function. The labs stay normal. Yet the cell does not. If this gave you a new way to think about your biology, leave a comment below. I read every one. And if you found this useful, the single best thing you can do is follow the channel. We decode a new mechanism every day. Everything you need is in the description and in the first comment. Before you go, we built a free 2-minute biological wellness assessment that reviews 4 body systems and gives you a personal pattern score. The link is in the bio, in the first comment, and in the description. 2 minutes. It puts numbers to everything we just discussed. The work on autophagy and fasting has expanded significantly in the last 3 years. A 2022 study published in Science Advances used advanced imaging to track autophagosomes in living human cells during fasting. What they observed was not random. Autophagy targeted the oldest and most damaged mitochondria first. The cell was not indiscriminately breaking itself down. It was selectively removing the components that were generating the most oxidative stress. This has implications for mitochondrial health that go beyond energy production. Damaged mitochondria leak electrons. Those electrons react with oxygen to form reactive oxygen species, ROS. In small amounts, ROS are signaling molecules. In excess, they damage DNA, proteins, and lipids. The standard narrative is that aging increases oxidative stress because mitochondria become less efficient. But the mechanism may be more specific. Aging reduces autophagy. Reduced autophagy allows damaged mitochondria to persist. Damaged mitochondria increase ROS. ROS accelerates aging. A 2021 paper out of the Max Planck Institute tested this in engineered mice. They genetically enhanced autophagy in one group and left the other as control. Both groups aged, but the group with enhanced autophagy maintained mitochondrial quality for significantly longer. Their cells had fewer damaged mitochondria, lower oxidative stress markers, and better metabolic flexibility at 18 months than the control group had at 12 months. The question then becomes whether humans can achieve something similar without genetic modification. The answer is time-restricted eating. A 2023 clinical trial published in Cell Reports Medicine assigned participants to either an 8-hour eating window or standard eating patterns for 12 weeks. Both groups ate the same total calories. The time-restricted group showed a measurable increase in autophagy markers in blood samples, improved mitochondrial respiration in muscle biopsies, and lower circulating markers of inflammation. Same food, different timing, different cellular outcome. Another angle emerged from research on protein aggregation. Neurodegenerative diseases like Alzheimer's and Parkinson's involve the accumulation of misfolded proteins that the cell cannot clear. Beta amyloid plaques in Alzheimer's, alpha-synuclein in Parkinson's. These are not foreign invaders. They are normal proteins that folded incorrectly and then aggregated because the clearance system could not keep up. Autophagy is that clearance system. A 2020 study published in Nature Neuroscience showed that when autophagy was pharmacologically enhanced in mice with a model of Alzheimer's, amyloid plaque burden decreased and cognitive symptoms improved. The system was not treating the disease. It was clearing the biological debris that was driving it. And in humans, one of the most reliable ways to enhance autophagy is fasting. There is also emerging work on immune function. And a 2019 study from the University of Southern California demonstrated that fasting for 72 hours triggered a complete regeneration of the immune system in mice. White blood cell counts dropped initially, but when feeding resumed, the bone marrow generated new immune cells at an accelerated rate. The old, damaged immune cells had been cleared through autophagy, and the system rebuilt itself. The same research group conducted a small human trial with chemotherapy patients. Patients who fasted for 48 to 72 hours before chemotherapy had fewer side effects, better immune recovery, and improved treatment outcomes compared to those who did not fast. The hypothesis is that fasting protected healthy cells by shifting them into a maintenance state, while cancer cells, which cannot enter that state, remained vulnerable to treatment. This does not mean fasting cures cancer. It means the metabolic state induced by fasting changes how cells respond to stress. And that has applications beyond oncology. A 2021 study in Cell Metabolism found that fasting before surgery reduced post-operative complications and shortened recovery time. The proposed mechanism is that autophagy clears damaged cellular components before the inflammatory stress of surgery, giving the system a cleaner starting point for healing. One of the more interesting findings comes from research on circadian biology. A 2022 paper published in Science showed that autophagy has a circadian rhythm. It peaks during the biological night when the body is naturally fasting. When researchers disrupted this rhythm by feeding mice during their rest phase, autophagy efficiency dropped even when total fasting time remained constant. The takeaway is that when you eat may matter as much as how long you fast. This aligns with human data, showing that late-night eating is independently associated with metabolic dysfunction, even when caloric intake is controlled. The body expects a fasting period during the night. If you eat late, you suppress autophagy during the window when it is supposed to be most active. You're working against the circadian system. The research is also starting to differentiate between fasting durations. 16 hours appears to be the threshold for activating autophagy. 24 hours increases it further. 72 hours activates additional pathways, including stem cell regeneration, but longer is not always better. A 2023 review in Nature Reviews Endocrinology noted that prolonged fasting beyond 5 days without medical supervision can lead to nutrient deficiencies and muscle loss. The body begins breaking down functional tissue, not just damaged components. The practical zone for most people is 12 to 18 hours. That is long enough to activate autophagy without triggering the stress response associated with prolonged deprivation. And it is sustainable. You are not eliminating food. You are consolidating it into a narrower window, and which allows the repair protocol to run daily instead of sporadically. What this framework reveals is that the body has two modes, growth and repair. They do not run simultaneously. When insulin is high, you build. When insulin is low and AMPK is active, you maintain. Most people spend nearly all their waking hours in growth mode, which means repair never gets its full window. The system was not designed for constant availability. It was designed for scarcity interrupted by abundance. And the repair mechanisms only activate during scarcity. The clinical implications are beginning to show up in human trials. A 2022 study published in JAMA Internal Medicine followed participants practicing time-restricted eating for 1 year. Compared to controls, they had lower fasting insulin, reduced visceral fat, and improved markers of cardiovascular health. But the most striking finding was not metabolic. It was cognitive. Participants reported improved mental clarity, better focus, and reduced brain fog. The researchers hypothesized that enhanced autophagy in neurons was clearing accumulated cellular debris that interferes with synaptic function. This connects to research out of Johns Hopkins, published in 2021, showing that intermittent fasting increased the production of brain-derived neurotrophic factor, BDNF. This is a protein that supports neuron survival and promotes the growth of new synapses. Fasting does not just clean the cell. It signals the brain to invest in resilience. The system interprets the stress of nutrient deprivation as a cue to strengthen, not shut down. The paradigm shift is this. Food is not just fuel. It is information. When you eat, you are signaling growth. When you stop, you are signaling maintenance. And if you never stop long enough, maintenance never runs. The body compensates for a while, but compensation has a ceiling. Eventually, the accumulation of damage exceeds the system's ability to manage it in the background. That is when metabolic dysfunction, neurodegeneration, and chronic inflammation begin to appear in labs. But those are late-stage markers. The process started years earlier at the cellular level In the gap between growth and repair, standard medicine does not measure that gap, but the biology operates there constantly. And understanding the mechanism gives you the ability to intervene before the damage becomes pathology.